Thank you, Professor Anand, and it's a pleasure being here again. This is the third time I'm speaking at the IVC and this time at IVC 2024. Thank you, Christian team, for the opportunities to speak at this occasion. And good morning, everyone. And good afternoon to some of my peers in India as well, and China. So I've taken this topic because I feel this is quite interesting, and this is something that is going on uh, since the SARS infection of COVID-19, as we call, and people are thinking seriously about taking this forward. And I can see some of the funding agencies as well putting in as much as 57 million US dollars to take it up as a in a global way, and trying to come up with some just other than the usual route of administration of prophylactic vaccines, like the nasal or any other way, so that we are prepared for a pandemic that may come, which I believe I don't think we will be seeing anything soon. Anyway, I thought a human challenge clinical trial, which is generally done for infectious diseases, give a background on that. All right. So basically, what the human challenge clinical trial, in other words, is also called as controlled human infection model. And this terminology is also used in some countries, like we have this terminology in India as well. And as the name says, or those four words say, human, which that's why we call it a clinical trial. And challenge is of the organism to the volunteer who is consented for this study. It's also called controlled because it is the challenge organism is a controlled one, and we always do ascending or that kind of study in terms of viral load or in terms of the vaccine itself. Yeah, and this is a model that people have seen that, over the last several decades. People have noted that we can have a good model to or a study to look at the way we can understand or investigate the onset and development of disease in a controlled manner, not only just with the organism, but in a controlled environment. Yeah. So basically, it's a intentional or a deliberate exposure of healthy human volunteers to infectious microorganisms. And these can be viruses, which is commonly seen or used as in this model. Bacteria is second in the ranking, and the third one we have parasite. That's only with the malaria, and fungus. Yes, we do not have a. People are working on that, but we still do not have a prophylactic vaccine for fungal infections as it is now. Anyway, so it's to tell it again, it's a intentional or a deliberate exposure of healthy human volunteers to infectious microorganisms, and do what? To investigate and understand the onset and development of disease in a safe and highly controlled environment, observe and analyze complex interactions between the microorganism and the human immune system, and also identify ways to disrupt and block an infection. Now, who are these humans? So they are the ones who are healthy, informed, and consenting. And if I may add, yeah, they are adults, because generally we do not do it in children. Yeah, so they are healthy adults who are informed and once they take a decision, they consent, and then they are safely exposed to an infectious agent of what we plan to use. Uh, and they will spend the duration of the study quarantined in a controlled but comfortable environment where they are continuously monitored for symptoms. The infectious agent or the challenge organism may be close to a wild type and pathogenic, but as I said, it's close to wild type. Now, why it is important? Because if you have a wild type and you do not know much about how to control them, then we are actually, first of all, we are deliberately exposing this organism. And if it's a wild type, then it is very difficult to control how you're going to treat them if the vaccine does not work out. Yeah. So generally these are close to wild type, and of course they are pathogenic, or they could be adapted or attenuated from wild type so that there is a less pathogenicity, and it could also be a genetically modified organism. HCTs, as a short for Human Challenge Clinical Trial, these have been conducted over the last 200 years, and they have contributed to to science with immense knowledge, so that we are able to understand the infectious disease, and thereby there have been quite a few development of many prophylactic vaccines and drugs. Now, out of those 30 pathogenic organisms that I can think of, there are about 19 of them that have been resulted in the development and commercialization. So going back to history, so there were attempts in 1774 by an English farmer named Benjamin Jesty, who is just a farmer, but he saw that when you can infect the family members with cowpox, and he could prevent smallpox from them getting smallpox. And sometime later, about 20 years later, there was an English physician and scientist, which I think we all know, attending this conference. It was Edward Jenner, who took the vaccine virus from milkmaid, uh, the maid who generally milks the cow. So this was a lady called Sarah Nems, who was milking a cow which had a fresh cowpox, and then Jenner inoculated, this matter from a small, uh, you know, into a, into the child who was 8 years old called James Phipps with smallpox. So what he did was he first took the matter or the pus from the cowpox from the cow, and then scrubbed it into James Phipps. And a few days later, because he did not know exactly what would be the time period, but fortunately for him, he did it after 2 or 3 weeks, and then he inoculated smallpox. So the matter which he scrubbed onto the arm of James Phipps, could activate the human immune system, and thereby it could protect uh, James Phipps from getting smallpox later. Because he saw that those milkmaids who were milking the cows which had cowpox, they did not get this smallpox infection at all. And that's what Benjamin also saw. So basically it's, and if you see most of those discoveries that have happened across, it was a keen observation of these two people that led to the development of smallpox vaccine later. Now, the other thing that was common here was, in addition to the observation, is there was no systematic methodology to induce the disease safely and reliably in humans, yeah, like we do now. Okay, so it was just a very crude method that we can think in the current situation. Second, there was no preventive or therapeutic intervention available for the infection. Now that sounds a little bit dangerous, because if you do not know or if you cannot handle the infection that you have been injected with, then it could lead to death as well. right? So, but anyway, luckily they did this, and if you see again some of the history, that was or the studies that were done in Cuba, Italy, Hong Kong, and even during the Second World War, where people were, I don't know how much they consented, but they were given, like, for example, he actually, had about 100 attempts to transmit yellow fever through, through the Aedes aegypti mosquito, and trying to bite in a healthy individuals. But people did not listen to him, and he also failed to show the results in different ways or in different setup. But 20 years later or 2 decades later, he was successful, and people realized that yes, you can transmit the disease, especially if they're carried by mosquitoes as well, like in you can see in malaria as well. Okay, so well, in Japan and Germany and many other places, people did not even take consent. They just, because they were prisoners of war, they just forced them to participate in this kind of studies. Of course, by doing this, one thing that we could learn was how a disease progressed from the start to the end. Okay, that's one part that we could learn from this, because when you know the pathogenesis of any particular disease, then it is easy to intervene at a certain point, whether it is within the body or in vitro. Like, we can come up with a vaccine as well, because we can then allocate which is the part that can be silenced, like for example, the S protein on a SARS-CoV-2 virus. Yeah. So that's what we saw over the last 200 years. Now, so there has been a long history in medicine, and I have contributed vital scientific knowledge that has led to the advances of several prophylactic vaccines, such as the first one that US FDA approved was cholera, for cholera. Then in 30 years of research, we could get a malaria vaccine last year. And then we had typhoid, Shigella, tuberculosis, and other prophylactic vaccines as well, and even some of the drugs. Yeah. Nevertheless, such research can appear to be in conflict with the guiding principle in medicine to do no harm. Anyway, we'll talk a little bit later about the ethics part over the next few slides. The first US FDA approved product based on the HCT was the Vaxchora, which is an oral cholera vaccine, way back in May 1998, followed by therapeutic oseltamivir for influenza. And then there was for RSV recently. And WHO had also approved the VI polysaccharide tetanus toxoid conjugate vaccine for Salmonella typhi. So that was part of their pre-qualification, and because of that, it was there. And this is this product that I was also involved with when human challenge clinical trial was done by the University of Oxford way back in 15, 16, 17, those days, in a smaller way, because we were the ones who gave this product for in one of their arm. There were 3 arms. So one of the arm was BI tetanus toxoid conjugate vaccine from India. So the next one is, when can a HCT be done? Although HCTs are not a required for every vaccine development program, there are reasons why a vaccine developer may want to conduct a challenge protection study in humans, which also can be conducted in animals as well. But let me tell you what are the problems with animal models, which, so there are, I've given some examples here, because we always think that animals are quite similar to human. But here are some examples which can be contrary. For example, tamoxifen, which is pharmaceutical developed by AstraZeneca for breast and cervical cancer or ovary cancer. It is also used for many other cancers as well. It caused or showed liver tumor in rats, but it did not show in human. Of course, most of the time, what we do in the pharmaceutical companies or in the industries, when we see such kind of thing, we just dump them. Now, imagine if this was dumped based on the results shown in the rats, and we would not have had tamoxifen in the market at all. And this has been doing quite well in the treatment of cancer. Yeah. But same liver tumor did not show up in the human studies. The other one, which has been very popular and it made a revolution in the treatment of leukemia, especially hematinic in chronic myelogenous leukemia. It showed it caused liver damage in dogs and other species of animals, but not in human cell assays and not even in clinical trials. So the other one is the prazoles. Prazoles, they showed low oral bioavailability in animals. So when we have this lesser, concentration of the drug in the plasma, generally what we do is in a human clinical trial, we, start with a higher dose. So when we start with a higher dose, remember this can be toxic to humans as well, because just for the data that it was showing less in animal blood, and then we start with a higher in human, it could be very, very difficult to manage. Yeah. And also at the same time, remember, we also have penicillins and aspirins in the market, and they was no HCT, and the people didn't even bother about conducting even in animals as well. But we have that drug, which has been successful over a period of time. Yeah, not necessarily that it was good enough. Yes, it was luck. Like the same thing with Edward Jenner as well. And not every organism or the vaccine that we take from a virus or a bacteria should have to show any cross neutralization with other organism, not necessary. So most of the time we have, some of the vaccines which are very, very particular for a particular strain, particular for a strain itself, not necessary against other mutations of the same organisms. Yeah. So this was a luck even with Edward Jenner, and here also we have some examples. Now, so the second part is the many organisms, or the even including the challenge organisms also, they are unique to human species. Like if you see, generally, if you compare with the stage 5, I mean, if you think about stage 1 is in animals, stage 2 is within animals, and stage 3 is animal to animal, and 4 is, in human, and 5 is human to human, transmission. So if you see human to human, we have only 5 or 6 diseases like the MMR, measles, mumps, rubella, smallpox, syphilis, and falciparum malaria. Okay, these are the ones which are human to human, which means we need to not just concentrate on this human to human transmission. We should also be concentrating on those coming from the animals to humans as well, or within animals to animals, and they can transmit just by bite out. Yeah, so that's another thing that we have to think about. And remember, human challenge clinical trial involve deliberate infection. So we actually give the challenge organism or expose them to expose a human volunteer to a challenge organism, and by that, we are able to precisely track disease manifestation, the recovery, and also the treatment in a controlled setting, not just in the wild where we cannot guess how the pathogenesis is on, because some may be already infected and might have some kind of coverage from a mild exposure, or some of them might be on some kind of treatment, which can also be cross-neutralizing the other bacteria or virus. Okay, so in that case, because we are, we have a strict eligibility criteria, and make sure that we include those volunteers who were never been exposed in the past by doing like ELISA or PCR to see whether there are any kind of antibodies from the past exposure. The challenge organism, as I said, is close to wild type, um, not just wild type. Uh, Pathogenic, yes, but we can also have them attenuated or modified so that we have a less pathogenicity. And that's an important part of it. The next one is that is the challenge organism should be the one that has an acute onset, self-limiting, mild, and can be readily and objectively detected and expected to clear the infection without treatment also in some cases. But most of the time, we always use a challenge organism where we can control the infection that comes up after exposure. Yeah, that's important because if you cannot control, that can be a very tricky situation and death can occur. But in some cases, like RSV, influenza, human rhinoviruses, and even with the SARS-CoV-2s, fortunately or unfortunately, there is no treatment available. And if you are using it in the right population, then you may, since there is no treatment, you could use in right population so that you don't have to worry about death. That can be a complication out of this study. Yeah. So most of the time we do it in we do this kind of studies where a proven treatment is available. The third part is the immune response and HCT. So we need to have a clear understanding of the pathogenesis and immunity to the organisms required in order to guide decisions and what type of or quantity of immune responses that you may see. Of course, we are born with the innate, and then we develop the adaptive ones. But we need to also see whether they would release the IgAs, E's, or M's and G's and things like that, and how much it can do. Because the next one is the correlative protection that I want to talk about, and then you'll understand that why it is important for us to know how long this the M's and the longer ones like the IgGs will be around so that we might have a protection for a period of time. Yeah. So with this immune response and the study on that, it can give us an insight into a vaccine design for further studies in a phase 1, 2, or 3 clinical trials in future, or it could be a vaccine efficacy or even a vaccine effectiveness studies that you can think of. Okay. So if you see what is the correlated protection, it is a specific immune response to a vaccine. Now, to make it simple, let me give an example of a pharmaceutical. Now, when you have a fever and it's an adult, we always take paracetamol about 500 milligram. Yeah. Now, would you take a 60 or a 70 kg person? Would we take 250 milligram paracetamol? Would it work? No, it does not work, right? So it means to say that you need to have a minimum safe therapeutic concentration in the blood to see a effect, or we say efficacy, right? Whereas in same thing in relation to vaccine, we think about immunogenicity. Yeah. So correlative protection is nothing but that determines the minimum amount of neutralization or neutralizing antibodies that are available for your protection. For example, in hepatitis B, the ELISA antibodies should be at least 10 milli international units per ml. Now, if it is below this, after 2 or 5 or 10 years, that means we are going to be exposed to that particular disease in future. Yeah. So this also has to be noted, or and also we have to think about the same thing with measles also. So there is a minimum concentration of antibodies that are required to give us a protection. So we call it as a correlate of protection. However, sometimes we cannot even know the true correlate of protection, and sometimes it is unknown, sometimes it is difficult to measure. So in that case, we always use the antibody measurement, what we call as a GMTs, geometric mean titers, and this suffice as a predictors of protection by a particular vaccine. Yeah. So with this, whatever we know, we can confirm them in the future phase 1, phase 2, or phase 3 studies that we can do. Yeah. The next one is the consenting. So as I said, we are exposing a healthy adult into these studies. And if I give an example from my experience of what the University of Oxford did, or the Oxford Vaccine Group did for the typhoid study, they sent out letters to about 100,000 people, and only 200 responded. And out of the 200 who responded and screened, only 112 were enrolled into the study. So you can see, just 1% of the entire population who has sent out those letters for asking whether they would be interested in a SCT, only 1% were eligible to get into a study. Yeah. But the main thing is they have to be informed. If they are informed and they understand the risk of what they're getting into, then they um, stick around. But they have to be told that this is an acute infection that may be caused, but these are manageable, which also means that they will have to stay in the facility for a period of time, and they need to focus on that also, because sometimes in a clinical trial we send them home, but when we ask them to come back for follow-ups, they usually disappear, which could be very risky, because being a medical person, I always say that when a person is being administered with the investigational medical product, whether it's a device, vaccine, or pharmaceutical, and they disappear at the third visit or fourth or fifth visit, I always take them as a worst case scenario that they are dead because of the product. Yeah. So here also the same thing. We don't say send them, however, but they are constantly monitored throughout their stay. So based on the disease, then we can take a call of how long they have to be in the facility, and also how we can manage. For example, cholera, is manageable with the fluid and electrolyte replacement. As simple as that. Yeah. What do we look for when we do the study? The first is from the infection that we have, we know how many days it will take to show their first symptom. Yeah, that is in the wild type and also in the general environment. But we will see and compare what about how long do they take to show their first clinical symptom in a controlled environment. That's one. Second thing is the viral load per ml of blood. Yeah, that's another way of looking at it. The third, in case we don't know what the antibodies are, we could always think about the immunogenicity titers as well. Of course, when we involve a clinician into this, they get an idea of, in a general public, how do they react? How would a COVID-19 react in a general public? And also you can see how do they react in a control setting. Of course, so we need to have that kind of comparison as well in individuals of similar age also, you know, because when we are taking healthy, it is fine because they do not have any comorbidities, but in general public, you could have a different age group, maybe elderly with several. Yeah. So whether those vaccinated have a different response also, that also could be seen because natural, someone who's exposed to a natural infection versus a vaccine-induced um, infection as well, and the response to that, we could also compare. And also for the adverse events, both the short and the long ones. Yeah. So, in terms of digital framework, remember in some countries, the challenge stock is not itself considered to be a medicinal product, which means good that we do not need to take a regulatory approval to conduct the study. However, some countries like the US and Europe, they take the challenge stock or the organism or the vaccine as a biologic, and that's why it is heavily regulated. So we need to confirm or conduct the study according to that regulation. But in some countries, it is not. The last one is a facility. So the investigators need to be qualified, definitely, but it is always better to have them in a medical hospital which is attached so that we can have the intensivists, the physicians, the neurologists, or cardiologists, and anybody else, so that they can be rescued and a top medical attention is being given to them. So it's better to do it. And I say same thing with the phase 1 clinical trial as well. Here also same thing. These facilities, that we use, and I can in the next slide I'll give you some examples also, or the facilities that are available currently around. So these facilities provide continuous monitoring and medical attention at appropriate points in time after the challenge organism is been exposed. Also, they need to have immediate access to the medical care and treatment. And in addition, the staff, including the janitors and even the administrative staff, which move around in the facility, they should also work in a personal with the PPEs appropriate for the pathogenicity of the challenge organism, not just a mask. Yeah, sometimes we may need to have those PPE suits. And also we have to think about the potential hazard to the environment also. Yeah. So we have to also think about the prevention of the spread of the disease, because once the challenge organism is been injected or administered into a volunteer, then they may be sneezing or coughing all around and spreading it to others as well. So we need to also think how they can be contained within that facility and not just sending them out of the facility. That's also. So some people will use the isolation pressure room, airlock, air curtains, all this. And also, if it is a attenuated or a wild type organism, it is likely that it can be restricted within that. Uh, for things like, administration of this challenge organism on a through intramuscular intradermal, maybe you can have a bandage around them so that they are not exposed if at all there is any infection of the administration. Here is a link for the Center of Vaccinology, in the Dalhousie University, Canada. Uh, I'm not going to click the link. You can always go there. This is the one facility which shows videos and photos about the facility, so you can get an idea about. Yeah. So these are some of the facilities that are available that you can think of. Mostly in the US, UK, um, and Belgium as well. Yeah. And also, as I mentioned earlier, Imperial College is taking a lead, and they have formed a ICVAC, a group where a lot of researchers are working on human infection challenge studies. Yeah. All right. So when is let us see when we cannot think of not conducting a disease, this kind of SCT studies. Remember, it takes time. So if you don't have time, patients, then it's very difficult to do this study. So it requires about a year or two. And I remember one of the influenza human challenge study, they took about a year to even, finish with the writing and getting all the things in place. So like that. So we need to have about a year or two to do the background work before a vaccine can even be tested. And also, during a pandemic, and if it's a dynamic pandemic, then we have difficulty because new variants may start popping up every few months, and it's very hard to use any of this challenge organism, because when you develop that particular vaccine, it may not be effective for another mutated variant. Yeah, that's another thing. The third is, if you know that the infectious agent or the challenge organism is known to cause severe illness or death in the absence of an rescue therapy, then we have ethical issues, right? So definitely people would not allow us to do this kind of SCT studies. It is absolutely impossible to run a Ebola human challenge trial because it has a modular rate. There is no way to predict who will have a severe disease. The same thing was with the coronavirus also, some of the vaccine constructs, but because they showed, a fatality in animal models. So during that, so in such situation, it is very risky to conduct. However, people have done the vaccine construct, or they have adapted uh, the organism in such a way that they are less pathogenic. And since we are using younger adults above 18, there is a likely that they may not get into a severe condition. That's another point. And as I said, you cannot do it in children or in any vulnerable situation, population. I mentioned earlier that if the challenge stock is regulated, that is vaccines, then you have to do all necessary work as per the clinical trial regulations. And also remember that the challenge stock should not be causing mutation during the manufacturing process also. So it must be free of mutations and reliably produce infection in animal models also. That's also important to know. And also, you need to have the characterization thought well, and also see what model system in terms of symptoms, kinetics, shedding, and transmissibility. That's also important to know. Yeah. So, if the challenge stock is not regulated, then the question comes, what about GMP and GCP? Is that applicable? And because it is not regulated, when we want to do the study, would IRB approval required? Yes, most of the time IRB approval is required, irrespective of regulations. And if it is if IRB approves this, then this is an academic study. And the problem is, if this is an academic study, the regulators do not take the data or accept the data for marketing purposes. They do not do. So this is another problem that we need to think about as well. And also, there are institutions, who also like the Institutional Biosafety Committee for the occupational and environmental safety. There also they need to think about. Yeah, so that is another promise that we can expect it. This means not all diseases are suitable. From what I spoke in the first half, that this is also, you need to understand that not all diseases are acceptable. All challenge organisms are not acceptable for conduct of human clinical trial. Now, let's see what are the benefits of doing a SCT study. Yeah, first is the dose. So a challenge trial might be conducted to titrate the challenge organism in humans before using the challenge in a vaccine study in order to determine the proper dose of the challenge organism and eliminate those not worth advancing. Because if we know that the amount of um, challenge organism, maybe the viral load is not enough to trigger a response, in terms of causing an infection, then we will have to go higher and higher. But with this higher load, would it cause a fatal disease or infection? That's also another thing. On the other side, when you have a vaccine candidate, we need to also think about what would be the amount of the virus or the virus like particles or the subunits that you need to inject. Yeah. So, and I give an example of how animal studies can give you a wrong information also. Of course, we don't do a bioavailability with vaccines because you can never get it. Anyway, so that's one part, the dose part. It also gives us a proof of concept as to whether a particular vaccine candidate might provide protection or not. That's another thing that it can give. Yeah. The third is the study design. So based on the HCT, we could also think about the case definitions. We could think about endpoints on even other study design aspects. And I can give an example from my own experience that, when the University of Oxford did the study for us, I mean, not for us, our product was one of it. When they did that, they took a case definition of fever, typhoid fever, as something that is 38 degrees Celsius for more than 12 hours plus the bacteremia in blood. Yeah. So that is one. So when we got that results, it was showing us with a efficacy, vaccine efficacy of about 52%, both in the VI polysaccharide and also in the conjugate. But when we change that definition to Maryland challenge study, which says 38 degrees Celsius, that's all, not 12 hours, and a positive blood culture for Salmonella typhi, we could see that the vaccine efficacy moved from 52% to 87%. So that's why it's also, so HCT can give you an idea about how you can define your cases for inclusion into the study. Yeah, that's one way. It also generates, it's also for the generation of appropriate hypothesis to be formally tested in traditional efficacy trials. It also gives an idea about down or up selection of various potential lead vaccine candidates so that we can advance only the best to large pilot or pivotal efficacy trials and also eliminate those which do not work. Yeah. And again, if you see the last one also, COVID was one where it was pandemic, but how many diseases have we seen? Even endemic. The endemic was a little bit about with Ebola, but we don't see much of them. So sometimes, yeah, it is very tricky because when we don't have a disease around, how do we know that the prophylactic vaccine is protecting us for whatever time it is supposed to protect? So that's another problematic that we have. So HCT can answer that. Yeah. The other one is evaluating the impact of prior immunity also. And you saw the cases with the dengue where people or the kids who were, already exposed to a certain subtype of dengue virus. And when they were given the dengue vaccine, it killed many of the kids also. So that's also important for us to understand. We did a Zika clinical trial for a Zika vaccine, and we were so careful about the presence of dengue in them as well. So this also gives an idea when you do a HCT. It also supports for emergency use of an investigation vaccine. For example, we had this COVID-19 example, right? It also gives an idea about the post-licensure exploration of whether immunity following vaccine, if it wanes or if or when booster dose may be required for a durable protection. Let's see quickly about the challenges of while conducting a human uh, challenge clinical trial. The first and foremost is the ethics. Yeah, so there are arguments against the use of HCTs, and this is around, it's been, I mean, it's centered around the ethics of participant compensation. Yeah. And also the first thing is whether the risk and lack of personal benefit can be compatible with the Hippocrates principle of primum non nocere, that is first do no harm. Yeah, so that's also another thing, because they can inflict potential risk in the study population that we are taking them. Importantly, challenge trials add the risk of infective agent to the already substantial risks of the experimental intervention being investigated. The human challenge clinical trials have a checkered ethical history, such as the German and Japanese experiments during the World War II, the sexually transmitted disease experiments in the Guatemala after the war, and also the Willowbrook hepatitis experiments that ended just in 1972. Yeah, so we all know, have heard and read about it, and so there is a uh, controversial. That's what we think from the ethical perspective. Yeah. And we are exposing the young, healthy participants, and that's why. And as I said, in SARS-CoV-2 infection, the amount of viral inoculum needed to produce an infection and to test the vaccine efficacy, and that's why we could control the disease. That's one way. But these patients, not patients, these volunteers were given about US dollars 6,200 for staying in the facility for 2 to 3 weeks. First with the understanding for screening thoroughly, and also giving the challenge organism. And 3 weeks later, they are given a vaccine to see. Yeah. So 6,200 in from US standards, or even 4,800 pounds in the UK. It may look like a small amount, but if the same amount is seen in Indian rupees, it could be approximately about 500 20,000 rupees. So that could be a huge amount in India, or this part of the geography. The next one is the risk and challenging volunteers with SARS-CoV-2. We saw it, and if it is if it mutates during the study, that could induce severe disease. Yeah, that's another thing. But fortunately, the net risk was acceptable, and because it was a younger population, there is a low risk. But people who believe in strong ethical reasons, they may not accept, they may reject this kind of studies. In terms of short and long-term adverse effects, understanding the risk, there is a review done by Adam Pips and published in the CID in February 2023. They reviewed about 2,800 articles, 276 in full, and the number of participants were about 15,000. And most of these studies were based on Plasmodium species, influenza, and rhinovirus. And over 40 years, they saw only 0.2% serious adverse events and no deaths. So which means to say that the risks are very relatively very low. And this is another study which was done between 1947 and 2014. They call it as a human challenge clinical trial, though they say RCT. So it was 26 RCTs, 400 plus participants, mainly with influenza virus. And they saw that it was only 0.04 experienced SC and no deaths again. So in terms of risk, it is very low. The third point is the cost of conducting this kind of SCTs. And I told you about the 57 million that people are trying to put in money. So that can be very expensive. I talked about the challenge stock, and also I talked about sending out letters to 100,000 people, and 100 were accepted. And you need to have a GLP and GMP facility as well. The labs that are going to test or do the assays, they have to be GMP standards. And it is anyway expensive to do a toxicology and immunogenicity testing as well, because before we expose into the human, we need to have some kind of basic knowledge or understanding about the disease as well, and also the safety of the vaccine as well. In short, just to give you a flow, so we start with on the left side, you can see the volunteers who are not previously infected are in the study. They are given the sequential dose escalation of the viral challenge and also the vaccine dose, in terms of the content. Then we look for the clinical response that is not more severe than the one associated with the natural infection because of ethical reasons. And we also need to compare those group of people of similar age who have been infected naturally as well, that you can take it from the public domain. They will be in the facility for a week or two prior to the challenge for viral and serologic test screening. And once they are done and good to go, they are randomized to receive either after the challenge organism, they'll get the vaccine or a placebo or a control. Then we wait for time because we know that it may take about 3 days or 4 days or even 7 days, like for a typhoid, or in some cases about 0 to 14 days, like for SARS-CoV-2. So based on that, we keep them in the facility. And then you could also see the reverse way, where you give the vaccine first and then the disease, and see how they are, how they are prevented. Yeah. So that's another thing. But the last one is to make sure that we have rescue therapy so that you prevent any significant morbidity and no mortality. So in conclusion, the human challenge studies evaluate the safety and immune response, and also the effectiveness of new treatments and vaccines against many pathogens, including malaria, influenza, typhoid, and cholera. That's been very successful. There has been increasing use of human challenge studies to explore pathogenesis and protection, and to evaluate vaccines and therapeutics. Since 1980, more than 15,000 people have participated in human challenge studies for at least 30 pathogen models, including SARS-CoV-2. Although AEs occur, severe and SAEs are rare. A particular concern with respect to some vaccine constructs against coronavirus is that they may induce severe disease following infection, as has been reported in animal models for both SARS and MERS. Yeah. If the vaccine candidate enhance the disease, the HCT would provide much more rapid evidence to support stopping the testing of more harmful vaccine candidate with far fewer vaccinated persons than a traditional phase 3 efficacy study or immunogenicity study. HCT provides early proof of concept for vaccine efficacy studies, particularly for pathogens without known correlates of protection at reduced cost and requiring only a small group of volunteers. Many ethical and safety considerations need to be addressed for vaccine challenge studies, and assessment is dependent on the pathogen in terms of biosafety level, virulence, route of administration, and host species. However, SCT has made significant strides in improving the ethical and regulatory aspects and accountability of medical research. Risk mitigation measures typically include evaluating the participants' risk of disease. That's why we make sure you take the medical history and understand their previous exposure to the pathogen. Also isolating throughout the duration of the study, excluding participants with employment posing risk of spread. For example, with E. coli, norovirus, and Salmonella, we have to, we can rule out enrolling food handlers because we never know whether they are already a carrier or not, and the chances that if they are challenged with the organism, they may also get into carrier, become a carrier later. You could also include, exclude participants requiring birth control, and also assessment of cardiac risk. And this is what we learned from the SARS-CoV-2 or COVID-19 infection. And also assess based on the assessment of weight or BMI, you can exclude them so that the risk level is lower. So that's how you mitigate some of the risk. Through HCT, we can understand a disease during low transmission periods and be prepared for high transmission seasons. There are some guidelines that are available. The first one came out in 2011 by CBER from US FDA. WHO has released one in 2017, and recently ICMR, which is in India, they have also released the policy statement for the ethical conduct of CHIM studies. Finally, human challenge studies are a tool to accelerate the development of new vaccines and treatments of infectious diseases. Probably they are not meant for every virus, bacteria, fungus, or protozoa, but this is a necessary evil that we need to take care of because knowing what we are getting into and understanding them very well and mitigate those risks can take us into a better design and conduct of a city so that we can have a better product for control of any infectious diseases. Thank you so much for your patient hearing and the opportunity to speak to a learner gathering. Thank you so much.